Ask the question. Thank you very much for the introduction. I'm really excited to be here. I've actually never been to UCSD. Telling people at dinner last night, our first escape during COVID as a family, we went to a little place in La Jolla, which is like 100 yards from the hotel I'm staying. So it feels a little bit like a homecoming. Thank you all for being here. So I have two stories I'd like to tell. The second one's optional, so we'll see how far we get. But broadly speaking, my lab is really interested in the mechanisms and consequences of mutation in sperm and egg, but also in somatic cells. But the story I'm going to talk about today is really about mutation in sperm and egg. I don't think I'm telling you anything new. Mutation is the primary source of genetic variation. It is the substrate for how creating genetic novelty, which allows species to adapt, and within an organism allows cells to have a competitive advantage or not with respect to other cells. And as many of you know, I know several of you work on this, over our lifetime, even though we inherit a diploid genome from our parents, that genome evolves over time within tissues as a consequence of exposure to mutagens and because of replication. So imagine you have a population of cells, mutation occurs in individual cells, those cells divide, so those mutations tend to propagate. If the mutations have a competitive advantage, they can lead to positive or negative selection, depending on the fitness effect of that mutation. So you can have an increase in the frequency of a mutation in the cell population or a decrease, and that can be because of selection or because of drift. And often in evolutionary genetics and even in mutation dynamics within in a disease context, we're trying to disentangle whether the prevalence of the mutation is the consequence of selection or randomness. And so the thing that I like to think about a lot is the mutations that we actually measure in any context are the result of a balanced equation in some ways. There's damage that arises. A subset of that damage is repaired. The unrepaired damage is what we measure. And so the measurement part, finding mutations, is also the challenge. And that's a lot of what our community and probably many of you work on. It's what I still work on. And so, when we're thinking about mutation, I think of it as this fine balance. This is one of my favorite books of all time from Rohinton Mystery. Has anyone ever read this book? I highly recommend it. It's an amazing book. But on one hand, if you have too much mutation, it might lead, or mutation in the wrong place, it might lead to disease. But we need a certain amount of mutation to form the substrate for adaptive evolution. So my lab, broadly speaking, thinks about how mutations arise, what are the mechanisms, when they arise, developmental timing, and what consequences that may have on phenotypes. What types of mutation? I think most of us think about spelling mistakes, single nucleotide mutations, but repeat loci expand and contract and breathe over generations. We have chromosomal rearrangement, structural variation, all sorts of stuff. But the challenge remains a challenge, but it was an extreme challenge, a decade ago. But thanks to technological advances, we're really good now at finding where mutations are, but I'm going to touch a little bit on some of the challenges that remain. So what my lab, this is kind of our graphic to represent what we do most of the time, which is basically writing code to scan through genomes in search of true mutation, like maybe there's a C mutation in this part of the genome. You might not be able to see it in the audience, but the graphic is a big DNA sequence in dark blue. But often what we find are false positives, things that are actually sequencing errors that masquerade as mutations. So our computational and sort of analytical challenge is distinguishing the signal of this real mutation from really high abundant noise of sequencing error and damage to DNA that arises during DNA prep that fooled us into thinking it's a mutation that's relative to the phenotype that we're studying. So this reminds me of kind of where we are in AI these days. Probably everyone has used, should I check this? Oh, it's from Rohinton Mystery. It's a fine balance. So probably everyone has used one of these AI tools to generate an image. Has anyone not done that? I'd be shocked. See no hands, so that makes sense. There's this really cool paper, I'll show you the link in a second, that talks about sort of the limitations of AI image generation. I think it's a nice analogy for the challenge we have in finding mutation. So here's 9 images. Can any of you guess which of the 2 of these are not real images? 2 of them, or sorry, 2 are real, 7 are generated by AI. Any guesses? It's intentionally not obvious. C1 is real? We got votes for C1? Okay. Any other guesses for what's real? Who guessed? Raise your hand if you guessed C1. Okay. I'm impressed. C1 is real. I stared at this forever and had no idea. C1 and A3 are real. The rest are not. And the reasons, there's all these little artifacts. So like B1 is not real because of this like little artificial visual artifact between the teeth. There's a merged stethoscope that I guess you could be able to pick out if you look really closely. Missing eye detail, so there's no iris, I guess, there. You know, a bunch of other stuff. And the basic point I'm trying to make is it requires extreme scrutiny. Like you could, if you had to want to do this at scale with an algorithm, you would have to like do some pretty intense image analysis to be able to detect this type of stuff. It's really extreme pattern recognition. And I think it's analogous to some of the challenges that we face in distinguishing real mutation in a sequencing data set from all sorts of artifacts that could arise in many different ways. So that's a lot of what my lab has worked on over the last decade, and we continue to work on. I'm going to talk about some of the technologies that many of you are familiar with to sort of amplify this signal to improve this signal to noise problem. Okay, so a lot of what we're going to talk about today is mutation detected in sperm and egg. I'm going to tell you a story about how that connects with reproductive fitness. And there, at a high level, there are 2 ways to study mutation in sperm and egg. One is to directly sequence gametes. I know people in this room have done this. It's a very challenging problem because most of you know that the mutation rate in gametes is on the order of 1 real mutation every 100 million nucleotides. The error rate for Illumina is something like 1 error every 1,000 nucleotides. So you have a 5 order of magnitude false positive rate there, a really, really difficult problem. Another way of sequencing or studying mutation in sperm and egg is what most of the community has done, where you collect DNA from mom and dad and kid, and you look for situations like this yellow mutation allele that's present in the baby, but not in either parent. And then you can figure out whether it arose on sperm or egg by linkage. You look at the reads and you ask, is there some other adjacent allele that's on the same read, thus the same haplotype, that we can unambiguously attribute to one parent or the other? And in this case, we can infer that that mutation arose on sperm because the linked green allele could only come from dad. Right. So the third option, 2A or 2+, is to do this in really big families. So I work at the University of Utah, and one of the resources that we have that my lab uses over and over again is these big multi-generational pedigrees where, in this case, there are 8 kids in the 3rd generation, but we have some families with 18, 19 children in a generation, believe it or not. And that's really nice for two reasons. One, with a 3-generation family, you can look for mutations that ostensibly arose in the 2nd generation and ask if they're real based upon transmission to the 3rd generation in Mendelian proportion. So you'd expect 50% of kids to have the allele if it's real. That's possible with small families, but because of binomial sampling, you could get 50-50 kind of easily. But if you now have 9, 10 kids to look at, you can actually look at really nice expected 50-50 ratio of that allele in the 3rd generation. I'm sorry for bumping into that screen. Right, so this is a common resource in the University of Utah, and they're part of this so-called CEPH cohort, the CEPH pedigrees. This is a French acronym for the Center for the Study of Human Polymorphism. This is one of the pedigrees. They were featured in a 1990s National Geographic. You can tell it's 1990s by the attire. So the 2 stories I'm going to tell you about today are a study we've been working on for the last couple of years. Shuvro in the audience here and the Alexandrov lab are helping us with a validation study of this work. And we're looking at elevated mutation rates in the sperm of subfertile men as compared to the sperm of fertile men. And if I have time, I may not, I'll tell you a story about some ongoing work looking at hypermutable loci in a 4-generation family now. We've now collected the 4th generation of these pedigrees. Okay, so first story, elevated mutation rates in sperm of subfertile men. I want to just first give a shout out to the people in the lab that have made this possible. The real star for this first story is Jason Kunisaki, who's in the PhD phase of an MD PhD. And it's really benefited from fantastic work from Tom Sassani, who was a grad student with me a long time ago and has come back as a staff scientist, as well as Michael Goldberg and Harriet Dashnow, who now has her own lab at CU Anschutz. Okay. So the setup for this is a couple of things. One, you may be aware of this, but sperm counts are declining in almost every country around the world. And there's some argument that it's controversial, but the argument is that the rate of decline is actually increasing, has since roughly 2000. And, there's a lot of, this is controversial, but there's a lot of explanations about why this might be. But, an example from this paper that I'm highlighting is, sperm concentration from a typically fertile male in 1973. This is meant to represent the average versus 2018. And at the same time, there's some epidemiological and now genetic evidence that subfertile men actually have poorer somatic health than fertile men. And I think about this as kind of related to the somatic theory of aging. So if there's elevated germline mutation rate, you might have elevated somatic mutation rates as well, which would lead to some of the observations of increased cancer risk, increased hospital visits, increased risk for many different phenotypes in subfertile men. So this background had us thinking about, well, could we actually demonstrate that there's elevated mutation levels in subfertile men, both in their gametes and in their soma? And so I just, with that background, I want to talk a little bit about the basic biology of sperm production and the many different ways in which subfertility can actually manifest. So with every second, a typically fertile man generates about 1,000 gametes. And those gametes, ultimately, even though there's millions of gametes in a typical ejaculate, it's only one that makes it to fertilize an oocyte, right? So why do we need such high sperm production, and how does that happen? Well, in a human testis, there's a bunch of these spaghetti-like structures called seminiferous tubules. And there's a cross-section of a seminiferous tubule depicted here. And basically, you have spermatogonial stem cells on the outside of these tubules. And as they commit to differentiation, they they self-renew to continue generating sperm throughout the lifetime of a male. But when they differentiate when they commit to differentiate the sperm, there's this maturation process that mature sperm end up being in the sort of the center of the seminiferous tubule. When they get to the center of the lumen of that, they migrate down the seminiferous tubule to the epididymis and then ultimately are ejaculated. And so over the lifetime of a male, there's spermatogonial stem cells are constantly dividing and self-renewing, and every cell division is an opportunity, right, for replication error and mutation. So, these haploid gametes that are eventually created in these seminiferous tubules are the result of many rounds of replication, ultimately followed by meiosis. And these mutations happen, albeit rarely, one mutation every 100 million nucleotides on average. So I think the question is just why are so many sperm necessary? And what is the kind of tipping point at which you produce too few sperm where you actually will be reproducibly infertile? And I think there's a lot to talk about here, and we could have a whole lecture just on sperm selection and conflict between sperm and egg across species and across evolutionary time. And I think it's an absolutely fascinating topic. But I think this graphic from this paper linked in the corner kind of reflects the extremes of sexual conflict that exist over taxa. Look at all the morphological differences among sperm from these different species. Everything from head morphology differences to tail morphology differences, how the nuclear package, the DNA package is formed and how that interfaces with oocyte, and all the different strategies for motility are honestly staggering to me. And speaking of sperm and tails and motility, it turns out that a typically fertile man, 40% of the gametes that they produce can't even move. They have mitochondria to produce the energy, but the tail doesn't function, or if they move, they swim in circles, or they just tire out. So it's a really small subset of gametes that have the morphological and modal capacity to facilitate fertilization. So beyond modal issues, we have all sorts of abnormal morphology. What I'm working towards here is hopefully quite obvious at this point. You need to produce a lot of gametes because most of the ones that are produced are not actually viable for fertilization. So one of the ways that infertility or subfertility manifests in men is just if you're producing fewer sperm, it reduces the likelihood that there's a single gamete that can make it all the way to fertilize an egg. And so the numbers game works out something like 10 to the 7th gametes are delivered, but only 100 to 1,000 actually make it to be close enough to an egg to facilitate fertilization. And of those, only 1 or 2 or 3, say, have the ability to have the right head shape and the right capsid proteins to make fertilization happen. So I was talking to folks about this at dinner last night. As I landed, I saw this article in the LA Times, believe it or not. So for sperm motility and the correlations between fertility and health have sort of taken its final form, I guess here. A team of 3 16-year-olds and a 17-year-old have started a company where they are racing sperm at a live event. This is going to have, I've never been to the Palladium in LA, but apparently this is going to be hosted at the Palladium and there's going to be betting on this. So people have developed microfluidic devices to actually race gametes. I'm really interested in this from a research perspective because then you can Then you can start studying like mutational load and really highly modal sperm versus immodal. But yeah, pay attention to this. I thought this was the Onion when it first came through, but it ain't. It's LA Times. Right. So I've probably beaten this horse to death, but ultimately, you know, sperm production is a numbers game. You need to produce a lot of sperm to be fertile because of attrition through morphology, motility, and just like endurance. And it turns out that sperm production is an extremely heterogeneous trait. There's this crazy paper from 2006, which basically had 140 sperm samples from one man, where they just measured sperm count over time. And you can see that for the most part, it's pretty consistent around 200 million sperm per mil, but it varies quite a bit. And this kind of reflects what the paper talks about is this reflects the extreme metabolic demand that sperm production requires. So this fluctuation could be a function of, you know, sleep, diet, exposure, all these different things, and just real-time changes and metabolism. So poor sperm production leads to male fertility issues. One of the things that is, I think, poorly appreciated is that male infertility affects roughly 10% of men. And despite misconceptions for many different reasons, that's a whole nother discussion. But it turns out that, in fact, 30 to 50% of infertility issues that a couple faces are male factor. Historically, I was always taught that it's like 80, 90% female factor. That's not true. So severity of infertility is a function of sperm production. So here's what this is a cartoon representation of what a typically fertile or normozoospermic male would look like, where we have at least 15 million gametes per mil. At the extreme end is the focus of a colleague of mine named Doug Conrad. He studies the genetics of azoospermia. There's no sperm produced without obstructive issues. There's basically there's no sperm production. So Klinefelters would be an example of that. In between are sort of tiers of this, where you have cryptozoospermia, so really low sperm production. And then what I'm going to focus on today, which is probably the most common form of male infertility, which is called oligozoospermia, where you're producing sperm, but at very low concentrations. Right, so that's going to be the focus. And there's prior work showing that genome instability in infertile and subfertile men is linked to sperm production. What's been seen clinically is kind of crude assays for DNA breaks. So you basically sample a whole bunch of gametes and you stain for double-stranded DNA breaks. And that intensity of double-stranded DNA breaks has been observed to be higher in subfertile men than fertile men. So building upon that observation, you know, we surmise that might be the result of defects in DNA damage repair, and should also manifest as an elevation of single nucleotide mutations. And we're interested in that because of the fact that men who are subfertile are more likely to undergo sample their sperm for IVF. And if there's elevated mutation levels in those gametes, that's something that's currently not appreciated and eventually could be tested for. So our basic hypothesis hopefully is obvious at this point. We hypothesize that sperm mutation rates would be elevated in the sperm of subfertile men compared to fertile men. So the University of Utah has a really large andrology clinic and sperm bank. So we've collaborated with a fellow named Kai Aspen, who's a close collaborator of mine, to study two cohorts, a cohort of fertile, or these normozoospermic men, and a cohort of subfertile men. And you can see that the subfertile men are below that clinical threshold of 15 million, reproducibly under that 15 million sperm per mil, whereas the typically fertile men are much higher. So our goal is to find mutations in distinct gametes. And, you know, the ultimate goal would be able to sequence the entire genome of a single gamete, compare it to the entire genome of another gamete and another and another and millions of them. We can't do that. Maybe that day will come. But what we can do is use error-corrected DNA sequencing technologies to find mutations that we believe came from one sperm. But the collection of mutations that we will find will be sampled from many thousands or millions of sperm that are sampled in the assay. And the way we do this is through a technology that probably most of you have heard of or use called duplex sequencing. This is a very fancy form of a broad class of sequencing approaches called error-corrected DNA sequencing. And the basic motivation for this is to overcome the sort of roughly 1 in 1,000 sequencing error rate of raw Illumina sequencing. And the way to do this is to use barcodes, in this case, to track each strand of a double-stranded molecule separately. You can then PCR amplify each strand so that you have redundancy. You can basically use a voting scheme. You can distinguish true mutations such as this green one coming from the top strand from PCR error or sequencing error, because the true mutations should be present on every copy. And so you can generate a consensus for each strand, and then you can generate a consensus for the double-stranded molecule. And the theory is that by doing that, you can drop sequencing error rates from on the order of 1 in 1,000 for raw Illumina sequencing down to on the order of between 1 in a billion and 1 in 100 million. And there's an ongoing discussion about what the actual error rate is as a function of the way the DNA is prepped and things like that. But it is a massive improvement over raw DNA Illumina DNA sequencing. So that is the technique that we've used. There's a whole other tangent that I could talk about how we designed our study and the approach we've taken. Suffice to say, we used a company called TwinStrand to do the sequencing. They're now out of business. And so we have a paper in review, and we need to do a replication study. And that's where my friend Shuvro over there comes in. And we're working with the Alexandrov lab to use their technique, which is called UD-seq, which is a genome-wide approach to doing this. So thank you, Shuvro. So the idea is that if you go from this sort of noisy, low signal to noise ratio with typical Illumina DNA sequencing with duplex sequencing, you have much higher resolution. So you can actually see the patterns that's there. Okay, so I think this is the first my data slide of the whole talk. So I'm sorry it's taken me 32 minutes to get there, but here we are. So this is a result from a paper we published 6 or 7 years ago, showing something that many of you appreciate, which is that for both sperm and egg, the number of mutations that are observed increase as a function of the age of the parent. So both eggs and sperm accumulate mutations with time. This left plot was from the pedigree study, where we're basically finding mutations in kid. We use nearby sites to infer whether it came from the single sperm or the single egg that led to that child. And what we know from those studies is that there's an additional 1.5 extra mutations per year for sperm and about 0.3 extra mutations per year, single nucleotide mutations for egg. Now, when we look for some of our cohort, the normally fertile men, we had 2 time points. We had a sperm sample. It's kind of a special cohort. We have sperm samples separated between 10 and 20 years for these men. And what you can see for the most part is there is an increase. There's a couple men where there's a decrease, which is a little surprising, quite obviously. But if you kind of average this out, we see an increase over time in mutation rate. So it's an additional 2.8 if all this is averaged. So that suggests that if we believe that the 1.5 is real, that's the real rate of increase. 2.8, that's maybe the residual is false positives. But the signal's there. So here's the main take-home figure, which we are really excited about and following up in many different ways. So in orange are subfertile men, and in blue are fertile men. x-axis is age. So again, we're looking at the rate of increase of mutation over time. And the y-axis is the calculated mutation rate using this duplex sequencing approach. And that you can see, what you can see for the most part is that there is this elevated mutation accumulation with age, but the y-intercept is very different for the subfertile men. The slope is statistically the same. The y-intercept is different. So this suggests at puberty, the starting mutation level in the spermatogonial stem cells potentially of these subfertile men is higher. So, you know, the question is, what is the mechanism that's leading to this? We've done all sorts of things to deal. Oh yeah, please.
Is there a compound in birth cohort here? So I noticed the age skew is very different. Normal.
Yeah, so that's actually something we're working with Shubro to address. It's really hard to get older subfertile men in our cohort because the way we find out about them is in Utah, you know, the age that people start having kids is lower than nationally. And so the recognition that you're subfertile comes earlier. And so these people are coming to the clinic because they're subfertile really young. So what we are doing in the follow-up work with Shubro is we've been able to collect more subfertile men across to do a better job with the age matching. So that's an ongoing analysis. So one of the other things that was new to me when we started this work is actually, I always thought that, you know, an ejaculate would be just sperm cells, but there's actually a high proportion of leukocytes and other somatic cells that are there. So one of the things that you have to do, and that has kind of plagued this field a little bit, is you actually have to go through pretty detailed protocols to eliminate somatic cells so that you're just measuring mutation in gametes. And so the lab that we collaborated with has kind of pioneered those techniques. And so we think that this result is robust to, you know, this isn't a difference of there's just more somatic cells in the sample from the subfertile men versus the fertile men. And so as I mentioned, we're working with Ludmila and Shubro to follow up on this, but I want to just walk through now is what we're thinking about as potential mechanisms for what might explain this. And we've spent a lot of time working through this and trying to eliminate potential models, but I think it's best explained by just walking through the fundamentals of germline development in the first place. So it starts with primordial germ cell specification occurs roughly around week 4 in human. Those primordial germ cells proliferate to generate a spermatogonial stem cell pool, which is quiescent until puberty. And that's the estimate. This goes back to work from James Crow, but in other organisms, it looks to be about the same. Roughly 30 cell divisions between primordial germ cell specification and the spermatogonial stem cell pool that's poised to generate sperm at the onset of puberty. So post-puberty, these spermatogonial stem cells do 2 things. They renew stem cells, but they also differentiate into gametes through this process, through the seminiferous tubules that I talked about earlier. So one model that we're considering is that there's elevated mutagenesis during the establishment of the germ. So this, I don't think Joe's here, but Joe Gleason has some really beautiful papers on sort of this early mutational load in the specification of the germ cell lineage. And so one hypothesis is that the rate of DNA damage or the capacity to repair that damage is worse in these subfertile men from the very beginning. And that might explain why we see 2 things. We see a mutation level that's higher at the onset of puberty, the extrapolation of that regression line. We see more mutations in the sperm that are there, and there are fewer sperm. Because if the mutation load is high enough, you can imagine some of those spermatogonial stem cells just dropping off and dying, no longer producing sperm. So that's one model. The other, oops, my animation's broken here. The other model is that, there's this, for the spermatogonial stem cells, there's this balance of self-renewal and differentiation. What if that balance is tipped a little bit, where in subfertile men, there's more renewal than there is differentiation. So more renewal means more cell divisions, genome replications, accumulation of mutation, and more renewal and less differentiation means fewer sperm. And then the last model is more of a meiosis model, where something in the final steps, as those spermatogonial stem cells differentiate into developing sperm, and they moved into the lumen of those seminiferous tubules. So there's something error-prone in meiosis in those individuals, or that leads to elevated mutation rate, and potentially, if that meiosis fails, then you see fewer sperm as well. To be honest, we don't really have any evidence one way or another about this. So one of the things that I would really like to do, and we've not been able to do yet, is actually look at the genetics of these individuals that have reduced sperm. So essentially genome sequencing of blood from these donors, so we can actually test the hypothesis that there's elevated loss of function or reduced fitness in the couple hundred genes that are involved in DNA damage repair. That's one thing we could do. Yes, please.
Any chance to do that in other banks, like GBSBC or something?
So we probably could. We can do it with our bank. We just don't have the money to do it yet. So that's actually a grant that we're writing right now. But yeah, it's a good question. So Matt Hurls has a really nice paper from UK Biobank looking at mutation level across all these trios, I guess. Maybe it was the rare disease cohort, I'm not sure. But yeah, they show individuals with elevated mutation rate, by and large, are rare, but the ones that they detected are men who underwent chemotherapy, and all their gametes have higher mutation rate. It's a really good question. It's something I really want to do, and that's a grant that we're writing now, but we've not looked at it at all, unfortunately. Okay, great. So the key takeaway is that, I mean, at least so far, we have support for this hypothesis that subfertile men have elevated mutation. We'll see from this collaboration with Shuvro and Ludmil whether that is manifest in UDSeq as well. But I guess I just want to remind people why we might care about this. I think this suggests that mutation rate is really a readout of spermatogenic impairment. It has obvious implications for the assisted reproductive therapies that men like this might pursue. It raises the hypothesis that if we studied somatic tissues in these same individuals, they would also have elevated mutation rates compared to fertile men. We had tried to do this in blood samples, and there's a weak and statistically insignificant signal for that. I think it would be an easier, well-powered study if we could look at more of a clonal tissue. I mean, blood is a mixed clonality. It'd be really nice to look at like non-sun exposed skin or something like that, but we don't have the ability to do that yet. And it raises the hypothesis that these men have DNA damage repair deficiencies, and this gets at some work that we did in the past with Molly Shaborsky, and that Matt Hahn has recently published. I don't know if anyone's read it, but he has this really nice paper that he calls the faulty male hypothesis. And just to summarize it, the hypothesis is that men have reduced, the repair of DNA damage in the male germline is not as efficient or robust as it is in the female germline. So my hypothesis here is that maybe this is a faultier male hypothesis, in the sense that it's even further reduced. The capacity to repair that damage is even further reduced. Okay, I have 15 minutes. I am going to try to just take 5 so that there's time for discussion. And I'll probably just talk for a minute about what we're doing, and then maybe I'll pick a few slides to show it. So the setup here is that my lab's obsessed with mutation, how it arises, what phenotypic consequences it generates. But and that's a passion of many people. But one of the things that is really underwhelming about the work that we've done as a community over the last 10, 15 years is that we're actually not even studying the most mutable sites in the genome. We're studying single nucleotide mutations, and most of those are at CPG sites where the cytosine is methylated. That has a 10-fold higher mutation rate than other single nucleotide sites. We've known forever that these short tandem repeat and variable number of tandem repeat loci are the most labile in the human genome and in all genomes. The challenge has been that we don't have technologies to study it. People here, like Melissa Jimrick, I don't know if she's here, but she's done some beautiful work, as you probably know, to use modern DNA sequencing technologies to characterize STRs. And it's not until recently that we have really high accuracy long-read DNA sequences such that we can span these very large variable number of tandem repeat loci to test mutation there. Okay, so what I think is just really exciting now is that our genomes are littered with these tandem repeats. So STRs, the motifs are between 1 and 6 nucleotides. So this is a trinucleotide or polyglutamate CAG repeat, underlies a bunch of different diseases. Variable number of tandem repeats are like, you have these 7+ base pair motifs, and they can also be compound. They're nasty. They have just this crazy composition of all these different motifs. And they're highly mutable, and they breathe. Between generations, they can contract and they can expand. And the size of the number of repeats is correlated with the age of onset for various diseases. What I just want to summarize is that we started a study a couple years ago where we basically threw the kitchen sink at one of our 4th-generation pedigrees. We use Oxford Nanopore, Pacific Biosciences, Element Biosciences, Illumina to basically characterize mutations in the entire genome. So we're looking at centromeres, telomeres, STRs, et cetera. And we can do this in 22 children across the 4 generations. And what these technologies allow us to do is to characterize all mutations in every one of these kids. And so that's probably obvious to all of you because you know sequencing technologies, but unlike short reads, if this is a TR locus in blue at the top, we need really long reads to span through it to actually measure how many motifs are in that haplotype. And so we can do this in each trio. So we basically can use these sequencing technologies to count the number of motifs. So let's say dad is heterozygous for one haplotype with 5 repeats. The other one has 8. Mom is homozygous for 7 and 7. And then we can use transmission genetics to basically figure out the haplotype that in this case, expansion occurred on. And do this for every single repeat locus in the genome and start to learn what are the cis factors in a haplotype that modulate the rate of mutation or the type of mutation at these loci. And so I'll close here. I just want to show this is a plot that we just generated 2 days ago. And it summarizes what I'm really excited about. And it's a complicated plot, so I'll take a minute just to walk through it. In red is the frequency of motifs of a certain size. So it might not be surprising to you that these short tandem repeats between 1 and 6 nucleotides, they're the most abundant in the genome. And as you get to these nastier and nastier motifs, they decrease in frequency. But what I really want to focus on is the mutation rate associated with these motifs of different lengths. So everything from so-called homopolymer loci, where it's like a run of A's, dinucleotides, trinucleotides. And what you see after length 6, so when we start to transition into these nastier repeats, there's this trough in the mutation rate. So mutation rate declines for short tandem repeats as the motif length gets longer, but all of a sudden it jumps back up again once we get into these more complicated and longer motif lengths. And so what we're working on methods to test now using these long read sequencing technologies is a hypothesis that, we think about these mutations as being replicative. DNA slippage on a lagging strand or replication that leads to an expansion or contraction in short tandem. But what we think we're seeing is a transition from largely a replication-based mutagenesis for the short repeats to more of a hybrid or more recombination-based mutagenesis for these longer. Because these alleles are really, really long. They're highly similar, and so are prone to ectopic recombination between the two haplotypes. So I'll end there. That is where we are developing methods right now to do that. And we have a follow-up paper from a paper that's about to come out looking at the mechanism behind these patterns that we're seeing. Yes, please. Yes. Oh, yeah. Thank you, Alan. Yeah, these are from, this is from that one pedigree. So what we're working on now is we've got funding to sequence 30 more of these 4th-generation pedigrees. And so looking at recombination, replication-based signals across all those samples. Right. So thank you very much for your time, and I really appreciate attention, and I'm happy to take any questions that you might have. Thank you.
Question?
So Fragile X full mutations only occur as de novo mutations, and de novo through the maternal germline. They don't occur through the father. So that would be presumably a recombination mediated type of event. Or are there other factors that might explain that?
Fragile X specifically, or recombination?
I'm thinking of a Fragile X example, but is it in every way identical to what you're observing, or is there something unique about it?
Yeah, so I think one of the things that we're working on right now, I'll try to answer the question this way, is looking at the cis factors on the haplotype that predict the likelihood of a mutation through the generations. So for each VNTR locus, we're calculating the mutation rate through the haplotypes in the generation as a function of how big is the allele in the parental haplotype and what is its motif composition. And perhaps not surprisingly, what we're seeing is the highest mutation rates are for the haplotypes that have the largest number of motifs, irrespective of what the motif composition is, just is there a lot of, so that suggests it's a better substrate for recombination. And then the other piece is what are the motifs that are most mutable?
Lilia had a question in the chat. Would elevated rate of mutations in subfertile men also lead to higher level of rare alleles that would lead to autism and other neurodevelopmental diseases? We know that parental age effect is known to be associated with ASD, and older men have higher mutation rate.
I guess yes and no. Yes, in the sense that if you have a higher mutation level, you have a higher likelihood that a mutation that leads to a, say, syndromic form of autism could occur. No, in the sense that it's not deterministic in that way. It's just a higher chance, higher mutation level, higher chance for a bad mutation to arise. Yeah. Yeah, one in the back. Oh, sorry. You're in control, Katie.
Thanks. Going back to your plots, I guess you don't have too many data points, but did you try breaking the the longer ones, according to kind of the types of the repeat units, like GC content
No,
or something like
that's,
that?
we are working on that, but it is it's a challenge with numbers. But yes, that's exactly what we want. To get it broadly, the sequence context and cis factors that drive this, yeah.
And so,
Great question. I wanted to share a slide on that. It's ongoing. So one of the challenges we had there is you can just, let me back up. What we need to be able to do that correctly is to not only detect that there's a mutation, we have to know what motif changed, and we have to know exactly not just which parent it came from, but which parental haplotype. So we can say, was it an increase or a decrease? And we just figured out how to do that like 2 days ago. But I would expect one positive control, and I always get this backwards. If Melissa's on, she probably knows. In eggs, I think it's more often contractions. As soon as I'm saying it, I'm sure it's wrong. It's the opposite. In one gamete, it's expansions, and one gamete, it's more often contractions. I forget which. So we expect to see that. But that's only been done for STRs, so we don't really know what it'll look like for BN2s. That is also ongoing. I would be shocked if there's not, because there is for STRs. The only repeat locus where there's poor to little evidence of an age effect is homopolymers. It's very, very weak. For dinucleotides and above, there's a strong parental age effect. So I would, but that makes sense under a replicative model. If it's largely recombination, I would expect it'll be there, but it could be weaker. If that makes sense.
Classic NAHR microdeletions are not as strongly followed.
Is that right?
In fact, some of them are paternal. I think Evan just had a paper showing that I think most NHR mediated microdeletion syndromes are predominant. Maybe it could be both. How was it? Was it age or just CCP is predominant? CCP1 microdeletion.
Okay.
Yeah. So that's a case where it's not father's age.
Got it. Thank you. Yes.
Thanks for the great talk. DNA methylation, is that something that you're able to look at? And then is the impact of it in terms of subfertile and fertile something that would be independent of the DNA mutations?
That's it. I just hosted head method earlier this week, and she showed some beautiful showing that in neurological disorders, when a repeat expansion gets stressed, these are huge repeat expansions, the allele is methylated, and that leads to because you reduce gene. And the question is, what mechanistically is the tipping point which the repeat gets big enough that it is like methylated like the repeat? That's something we've not looked at, but we can look at because with the PacBio data, we get all the methylation. We are complete noobs in terms of working with the methylation data with this, but it's something we're doing. But yeah, great question.
Well, I think that's it. Thanks everyone for coming, and thanks Erin for speaking.